Greetings. My name is Bismana William Gad, alongside Lucifer Mark Collins, Kasozi Mark Carlos, and Mascot Ian Says. And here to present a topic explainable meta-learner for predicting antimicrobial drug resistance in dynamic healthy care environments. With our keywords being long short-term memory, genomic sequencing, amino acids, antibiotic accessibility testing, and Oxford Nanopower. Antimicrobial drug resistance has been a very growing health concern due to prolonged and incorrect use of antibiotics. Particularly healthcare environments lack fast and efficient, affordable antimicrobial resistance prediction tools. The existing advanced ones take between 6 and 24 hours, which in this time, they lead patients to being given broad spectrum antibiotics. So several improvements have been made to these methods, and they have been accompanied by several computational problems. Inability to generalize to novel mutations and predict new mutations, massive size of genomic data. That's obvious because there is a whole batch of genomic data, complexity in resistance methods, lack of clinical relevant explanations to these. So our project therefore highlights the development of a machine learning model that makes use of meta-learning and recurrent neural networks modifications to predict antimicrobial resistance quickly and provide clinically meaningful explanations and to guide clinical decision making. Our project objectives are: one is to obtain compact and standardized representations of extensive bacterial genomes in a more usable format. Yes, this is done by implementing KEMA encoding. To design a meta-learning models for predicting resistance patterns in bacterial genomes and their associated profiles against antimicrobial drugs, and to also obtain clinically meaningful explanations for the predictions made in order to acquire insights into the relationship between genetic features and antimicrobial drug resistance. Lastly, is to analyze and convert bacterial genome resistance sequences into amino acids in order to establish a relationship between the two.
Thank you very much, Gad. This project offers an efficient method that employs meta-learning together with a long short-term memory recurrent neural network, while utilizing KEMA encoding that helps to generate smaller bacterial sequences that are easier to work with, together with explainable AI techniques to explain how conclusions were derived. In addition, we use a BioPython library for our Python-based amino acid analyzer to help determine amino acid relevance in specific resistance processes. So the recurrent neural network that we use is an artificial neural network that is inspired by the cyclical neurons, a cyclical connection of neurons in the brain, and we use it majorly for two reasons. And the first being that it accepts many different types and representations of data, as well as its ability to predict sequential patterns in the presence of sequential distortions. A limitation of these recurrent neural networks is that it cannot hold information for long. And that limitation was solved by the use of the long short-term memory form of a recurrent neural network that maintains a cell that acts as a memory, as memory to help track dependencies during training. The meta-learner that we are using works in such a way that different models generate predictions first, and those predictions are used as input features for a meta-learner. And that helps to provide a model with improved generalization, ability to combine multiple perspectives, and bias avoidance capabilities. And this also helps to provide a model that is able to perform predictions on novel mutations. For the explainability, we use Lime and Alibi whereby Lime helps to identify the specific segments that contribute most to a given classification decision, whereas Alibi provides rule-based explanations for why a model made a given prediction. So the data that was used was from the Comprehensive Antibiotic Resistance Database, which is a public bioinformatic database of resistance genes, their products, and associated phenotypes. And for this project, we used 3 FASTA files. The first FASTA file contained antibiotic-specific sequences, whereby each sequence that showed resistance was associated with the antibiotic to which it showed resistance. And the second file included genes whose overexpression resulted in antimicrobial resistance. And the third one included nucleotide sequences for non-AMR genes. So the first FASTA file was used to train the machine learning model on how to group and match sequences with specific antibiotics, while the other 2 FASTA files were used to train, test, and validate the models, as well as used as a cross-reference for the amino acid analyzer. So the data in its raw form presented several imbalances. As we see on our right, the original distribution presented several imbalances. So for model training and validation, all the classes that were underrepresented, whereby those that had sequences of less than 5 were filtered out and ignored, and those that had also very low sequences were augmented, and finally were able to achieve a data set with equal number of sequences to avoid model overfitting and a bias. Thank you very much, and let me welcome my colleague Ian to take us through the next part, which is the system architecture.
Hi, this is Masiko Ian Sezi. I'm going to carry on from Colin's presentation. So I'll take you through the system architectures first of all. So our data was acquired from the CAD database. CAD is the Comprehensive Antibiotic Resistance Database. It is an open database that provides genomic data concerning resistance. So it has a lot of data, but we were mainly interested in the antibiotic specific sequences, genes whose overexpressions led to antibiotic resistance, and nucleotide sequences of genes linked with antibiotic resistance. So this data was acquired, and then EDA was done, exploratory data analysis, to determine how balanced and how ready the data was for use. We realized that it wasn't actually very clean and balanced, so we employed some techniques like augmentation to bring the sequences up to speed and to drop the severely underrepresented ones. Additionally, we used the SMOT and smooth techniques to overrepresent the minority classes and underrepresent the majority classes. Augmentation was used to generate novel sequences based on the sequences we already had without changing the meaning and confusing the models. Kema encoding was done. This is an encoding technique where if you have A, B, C, D, E, F, and maybe have a K value of 2, you'll have subsets of A, B, B, C, C, D, DE, yeah, emphasis on the overlapping. So Kema encoding is employed because most of the sequences from this dataset do not have specified starting points. So usually when you don't know the starting point, you may confuse the model and end up with a completely meaningless sequence. So to cater for this, Kema encoding is introduced because with the overlapping and the different subsets, you realize that you've made up for all possible starting points. So on the left, after Kema encoding, is the amino acid analyzer. Like Collins has mentioned, this project is split into two: an amino acid analyzer and a machine learning model. So the amino acid analyzer basically was intended to prove the hypothesis of whether frequency of certain amino acids contributes to particular types of resistance. And it was done by getting the sequences and then grouping them into k-mers of 3,3 because antibiotics are made up of 3 nucleotides, and then matching them with the antibiotics and then arranging them according to frequencies of occurrence and seeing which ones occur most in given resistances. And then the machine learning models were basically recurrent neural networks, long short-term memory networks. We had them combined, then individually, then combined with the meta learner. So after all this, the two were combined. The meta learner was introduced into the rest of the application as an API. And lastly, the application implements explainability together with the machine learning models. They implement explainability using Alibi and Interpret ML and Sharp. And they also generate clinical reports and PDF reports detailing what has been determined and alternative medications to guide the clinicians. We also used we used TF serving for deployment, and the last bit shows a medic trying to use our application. So implementation details, like I described already, we used Python. Python was basically for the amino acid analyzer, and because we used Colab and Cargo notebooks, they are Jupyter notebooks and they use mostly Python. Yeah, so some of the libraries were TensorFlow, PyTorch, BioPython, and so on. All these are Python compatible libraries. So these were our results, some of them. So first of all, the recurrent neural network with KEMA encoding, this one had the least accuracy because it kept running out of RAM. KEMA encoding, unless done locally in a notebook, it keeps running out of RAM. So we dropped it for temporarily and introduced recurrent neural networks combined with long short-term memory networks, and these had the highest accuracy. However, the meta learner, after being introduced, we had to sacrifice some of the accuracy because we really needed the meta learner because it allows the model to make predictions from sequences it has not interacted with before. And since this genomic data is huge, we cannot feed all the data into the system. So we would need a technique that allows it to learn from history and the existing sequences and make future predictions. So the combined LSTM, RNN, and MetaLearner were selected as our best models. So lastly, here is a representation of some of the data generated from the application. So it is able to generate, this is a snippet from the PDF report it generates. So when you provide it with a set of sequences, it combines them and generates a PDF report depending on the specification, and it goes ahead to explain why particular results are the way they are, why which amino acids were overrepresented and why, and it also suggests alternative treatment methods. Thank you. My colleague Carlos will take you through the lessons learned and challenges.
Yes, this is Cosmo Carlos. Like my teammates are taking you through the introduction, the implementation. So I won't take you through the challenges and lesson learned. So among the lessons, we share different skills as a team, working together remotely, physically, yeah. And also we improved the, because this was a machine learning project, obviously, improved our skills in the sectors of machine learning, bioinformatics, and software optimization to come up with the APIs. Then also among the challenges, we faced the computational complexity of KEMA encoders and LSTM architecture, but data imbalance is among also a challenge. In the data set, the genomic sequences were not equally represented. Yes, and also other challenges like then also on volume feature work, we are planning to develop a cloud-based electronic medical systems record system to integrate it with that of come with, and also the APIs so they can be used by various workers. Then also for among the achievements, we had this week we came up with an AMR prediction project which was successful, and it was integrated. Like we included machine learning, bioinformatics, and data visualization to analyze bacterial gene sequences and predict antibiotic resistances. Like my friends have been taking it through how the system is going to be working. Then also for the conclusion, by addressing the growing threat of tuberculosis resistance, our solution provides early prediction. So according to the record you have, we see the way the genomic sequences of the patient, and know which know which antibiotic is being resisted, and know which antibiotic is being resisted, and provide a solution to it. That is basically what we are working on. And also, these are my team members, Ian, Lasiba, Collins, and Gad. Thank you so much.